We spend so much time on, somatic mutation theories, which is such a bad theory. There's so much wrong with it that I'm surprised anybody still takes it seriously.
Unfortunately, the National Cancer Institute takes it very, very serious. That's why we have no progress.
That's what I thought too.
Welcome to Target Cancer Podcast. This is Dr. Sanjay Juneja. I'm going to skip the intro because I am so excited and humbled that we have two of our guests that everyone's asked for multiple times because of this culture shift on thinking about what is causing cancer and can we do better, and what does diet and nutrition and metabolism have to do with its pathogenesis, how we treat it, when it recurs. And if you're thinking that I'm going to say this, yes, it's true. I have Drs. Jason Fung and Dr. Thomas Seyfried today to discuss the things that we've been hearing in the news, and seeing in the data about cancer becoming, a diagnosis younger. It's in higher volume. And if you have not listened to these podcasts before, highly recommend doing so, probably before this podcast, because it has really opened my mind up as an oncologist on not only what is causing it, but what should we be considering while we are treating it? Something called truncal mutations or root causes. So without any further ado, Jason and Thomas, thank you so much for being here.
Thanks for having me.
Yeah. Thank you very much for having me also.
We're in a world right now of this GLP-1 stuff, and everyone's talking about Ozempic and bringing, body fat reduction and obesity down. And we've talked a lot about how obesity and inflammation, in the past shows data on causing cancer, but also in Jason's podcast, especially about how insulin is a growth factor and kind of stimulates these cells to say, grow, grow, grow. Cancer is unregulated growth. I'd love to start on talking about, in an untreated cancer, how does insulin and sugar levels play a role in its, pathogenesis quickly? And then also, do you think that the pathogenesis will go down when we have agents that are able to better control our metabolic activity?
Well, no, I think these are important issues because what we're seeing now, I think this obesity issue is now seems to be replacing smoking as a major cause of linkage to, let's put it this way, a risk factor for cancer. And you're right, there's an insulin issue, there's a glucose issue, and the systemic inflammation in many people that are obese, and let's not include every obese person on the planet in the same basket. We know there are some obese people that are actually very, very healthy. They have unbelievably healthy blood work and all this kind of stuff. But that's the exception rather than the norm. And if your insulin is high and your blood sugar is high, and you have a tumor, it will generally cause that to grow faster. And I think that's pretty much obvious to those of us who understand this issue, but apparently not obvious to a majority, or I don't want to say majority, but to many oncologists who have never heard that glucose has anything to do with cancer, and often encourage their patients to drink Infamil or sweet things to maintain weight, which is just the opposite advice that you would want to give someone. So how is it possible that people treating cancer would not know that glucose and insulin levels elevations are provocative to driving the dysregulated cell growth? So, Sanjay, you are an oncologist. You are the voice of your profession. How do you explain this? Because I know Jason has spent a lot of time on many podcasts telling us about this, and apparently they might not have heard him either. So I'm not really sure how to explain this. Give me I should ask you that question. What do you think?
I think part of...
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
To be honest, I don't have an explanation other than you do internal medicine and then you go on oncology. So internal medicine, it's not necessarily a pre-cancer internal medicine training. We just learn about, the consequence on diabetes and how that has to do with, vascular stuff and kidney disease and stroke, endothelial stuff. And then you just kind of jump into the treatment of cancer, which is, predominantly immunotherapy, chemotherapy, replication cycle. And then now this whole excitement about targeted mutations or targeted therapies that both of you have kind of taken, you spoiled the joy of for me because, at first I was like, great, we have all these targets. And if it's showing blueberries, we can attack blueberries rather than trying to hack away the whole limb because that causes toxicity. And you're like, no, Sanjay, both of you, hack away the limb or go ahead and go at the trunk of the tree, but do so without necessarily poison. Because what we forget is that trunk of the tree actually requires things to grow, right? It has to be watered and it has to have some kind of fertilized ground. That's why hydrangeas never do well in my house because I can't even keep the constituents to make that thing grow, which further downstream are blueberries, strawberries, and I'm saying that as if it's a positive thing. But in these cases, the reason that the cancer is growing, we're chasing this all in targeted therapy world, and oncologists have a lot of pressure to make sure they're up to date with the standard of care of assessing way down on the limb what different things are growing and making sure that they're adhering and buying the molecular tests so that we can make sure the therapy attacks it. And that's why I think these conversations are so important. It is not in our repertoire or at NCCN to really think about, are these all just downstream problems because the ground in which the trunk and the roots exist are continuing to be watered. And part of it is because we believe, well, all ground has to be watered. So you can't really influence too much the glucose and insulin, which is obviously wrong.
Yeah, I mean, the thing is that you get all these sort of exciting things about, there's this mutation, and we're going to target this and this. So I think what happens is that you get the idea that cancer is caused by these genetic mutations, and it's a genetic disease, and it's just bad luck. And that feeds in, but it's actually completely untrue. Because when you step back and you say, okay, well, what causes cancer? And they've done studies on this. What is the biggest population attributable risk to cancer? And smoking's up there around 30%. But what everybody forgets is that way, way up there, as Tommy, you're saying, diet and obesity is also about 30%. And it completely dwarfs everything like, radon and, chemicals and chemical carcinogens and stuff. Right? It's a huge, huge risk factor, and that's all environmental, not genetic, right? So you have to look at the environment. And then you look at what you talked about earlier, which is that, hey, we're getting young people getting tons of cancer. And if it was a genetic disease, you'd say, the genetic makeup of the population has not changed in the last 10 years. Other than through immigration, right? But that's not what we're talking about. So what is it that has changed? And of course, diets have changed, obesity has changed, and the entire thing. So I think what has happened is because of the advances in the genetic, technologies and the treatment, you get this idea that it's 90% genes and 10%, but it's not. It's like, 80% environmental. A lot has to do with your diet and weight. And we know this because the World Health Organization has like 13 different cancers that are obesity associated. One of them, for example, is colorectal. And now you see this shift. Younger people are getting colorectal cancers and bad ones too. Well, why? Well, not so hard to understand. If obesity is a related factor, then there's more obesity. So therefore you're getting more. You're getting more obesity younger, right? Kids are getting obese in their teens now. So therefore, you're getting more. Just like you have more type 2 diabetes in young people now. Same thing, right? Pediatric clinics used to be 90% type 1 and 10% type 2. Now it's like a 50-50 type 1 and type 2, right? You get like 15-year-olds with type 2 diabetes. So this whole thing is, it does require this whole shift in thinking that, hey, this is not just a genetic disease. That's not to say that genes have nothing to do with it, because, and it's not to say that these targeted therapies aren't good, because they are good. Obviously, they spend a lot of money on it. But you have to, when you're thinking about how these things develop, you have to think about what is important. So one of the things, obviously, is glucose and insulin. And it's like, you look at breast cancer, and it's been known for like 50 years, okay, at least, that breast tissue doesn't really have a lot of insulin receptors. Why would it, right? It doesn't need it. But breast cancer has like piles of insulin receptors. Well, why? Well, because if it has lots of insulin receptors, then you have lots of insulin, then you can open up the GLUT4, channel, pour in the glucose into this cancerous cell, and it can grow, because we know, again, that it uses glucose to grow. I mean, Warburg talked about that ages ago. So it's like, okay, well, if that's important, then limiting glucose and limiting insulin is important in the development of colorectal and breast cancer, which for some reason just gets lost in this whole, it's all about genes, it's all about genes, it's all about genes. So we get so blindsided into this one sort of, view that we forget about everything else that's gone on. And even when you look at what causes cancer, like genetic factors, and they've done a big study on this, it's still a relatively small factor in causation of cancer. It's like 5 to 10% versus 30 for diet.
Yeah, and you both mentioned this in your podcast about how that was pretty much nailed on the head when we transplanted people from other parts of the world, because we said, wow, they don't get cancer at all, they don't get colon cancer, they don't get this, whatever the cancer was. We're like, they have some golden, TP53 that can't be hurt, like, or 8 of them, like an elephant does. And then when we brought them here, or the diets that were American were introduced to where they were, all of a sudden they started having cancer. And I'm saying it loosely, but literally transplanted them to study. They were eating American food and kind of much richer, different natured food, and cancer started to appear. I mean, that in itself was very memorable that both of you mentioned.
Yeah, and remember the story of Burkitt, right? So Burkitt was, he did a lot of missionary work, that's what they called it back then, in the 40s and 50s, right? So I think he was in Ireland or somewhere. And he went to Africa, and then he's like, hey, white people are getting all this colorectal cancer, but the Africans don't. They get like 0 colon cancer. But then when you take those African people and you westernize them, they start getting colon cancer, right? And that was observed like in the 50s, 40s, and 50s. So obviously the diet was a huge factor. Now, he thought it was all about fiber, and maybe it wasn't all about fiber. It may have been all these other things, the refined foods and all that stuff. They didn't have the same chemicals and stuff, but probably the stimulating the hyperinsulinemia, the glucose, at least played a role in it. But same thing, right? You take, and same thing in the North and in the Arctic. You had all these, in the 40s, you had people going up into the Arctic, researchers going up there. Why don't these native peoples get cancer? There's 0 cancer. Then, of course, as they start to westernize them and give them sugar and refined flour and stuff, and they start getting weight gain and all this type 2 diabetes, hey, their rates of cancer are actually worse than ours now, right? So it's like, well, obviously it had to do with diet, right? That's the key factor.
And what y'all, one, that's the cause, and 2, what y'all both mentioned in the podcast, Jason, you talked about, and I've never been the same since then, about DCIS and are we over-treating? Because we know, to your point, cancer, needs fuel, and community oncologists roll their eyes when they hear, well, doesn't it fuel cancer? But you need fuel to grow. Cancer is unregulated cell growth. And you were like, are we over-treating DCIS when we could have modifications to possibly temper it or not give it the gas it needs to go uphill, and then let our immune system, do what it does? And then Dr. Seyfried, you took that a step further and said, okay, even if it's invasive, can we limit the fuel or nutritional sources to car types that need something else. Electric cars need the charge station. So I don't have one. I probably need one to do a lot of driving. I have to use gas. And you were like, okay, we know that one of those, if the Tesla can't be charged, it can take away electricity. But that half and half, you're like, I want to be able to take on something additional. And went into these kind of amino acids or other kind of escape means to be able to process, quote unquote, fuel and energy due to their mitochondrial changes. So if we could talk about that for a second, mitochondria, how that gets changed and influenced with diet, sugar, insulin, and that strategy, Dr. CP, that you were talking about.
Yeah, well, I think to follow up on this with respect to the genes, and also to segue into what you just said, we have to look at the theory that's driving the approach, which is the somatic mutation theory of cancer. Now, for those who would think cancer is a genetic disease, they would say and argue that smoking and obesity and inflammation induce somatic mutations in the DNA. So it's the abnormal. Yeah, you have an abnormal diet. You have obesity is inflammatory, and inflammation can lead to DNA repair problems and these kinds of things. And therefore, even though we're seeing a linkage with diet earlier onset, it's still considered a genetic disease, because if we look at the tissue itself, we see all kinds of different mutations in the tissue. Even the young people, when you look at the tissue from the young people, or the Africans going from one, or the Inuits going from one diet to another, when you look at their cancer, it's loaded with all different kinds of mutations. So they say, well, it's a somatic mutation as opposed to germline mutations, which, like Jason said, it's the germline. So what we have done is we have explained where all those mutations and how they link to dysregulated cell growth. The somatic mutations are coming from the mitochondria itself. When this inflammation coming from obesity and all these other things, or from smoking, or from virus infections, or whatever, they lead to reactive oxygen species in the cells, ROS. ROS are carcinogenic and mutagenic. So they lead to the mutations that you see in the DNA. They break proteins. They disrupt lipids. All of these things lead to problems in the energy utilization and homeostatic state within the cell. But every cell in someone's tumor has a different constellation of mutations from any other cell. There's no all my tumor cells can be identified by having a singular kind of mutation. That's not true, and we've seen that over and over again. But what every cell in that tumor does share is a dependency on a fermentation metabolism, which is, in one part, the glucose is fermented to lactic acid, and this is what we see in all major cancers, an accumulation of lactic acid. And the second thing we see is accumulation of succinic acid, which is coming from amino acid fermentation. When the mitochondria become defective, the cell has to fall back and getting energy from someplace else. And that energy has to come from fermentation. It's called substrate-level phosphorylation. And in the cytoplasm, it's through the glycolysis pathway. And in the mitochondria, it's through the glutaminolysis pathway. And glutamine is the most abundant amino acid. So we have shown that glutamine is actually fermented. It's a fermented amino acid. Now, the mutations are accumulating in the cancer, different from every other cell in the tumor. So, but according to the somatic mutation theory, we'll try to target some of those mutations in the tumor cell with the hope of reducing, and you often don't find complete cures from targeting mutations, mainly because you have other cells in there that may not have those mutations, and therefore they bypass the targeting. And at the same time, now we're realizing that many of these driver mutations, which are different from passenger mutations, were the mutations that were thought to be the ones responsible for triggering the dysregulated cell growth. Now, when you try to target driver mutations, new information says and shows clearly from numerous studies that normal cells in our body and our tissues have these same driver mutations that are not associated with dysregulated cell growth. So when you use an immunotherapy or a targeted approach to managing cancer, yes, you may slaughter a large number of the tumor cells, but then you have promiscuity and you start destroying your liver and kidneys and some other organs that also may have that same epitope from the mutation. And therefore, it's no longer what we consider a precise targeting. It's an imprecise targeting. And what we do know is that in every single cell, cancer cell and tissue that we have found, they all have abnormalities, number, structure, and function of mitochondria. And our most recent paper shows that all major cancers accumulate cytoplasmic lipid drops in the cytoplasm. And those cytoplasmic lipid drops, triglyceride accumulation, is there to protect the cell from death because they can't use the fatty acids, so they store them in these vacuoles in the cytoplasm. And that's all due to dysregulated mitochondrial function. So if the mitochondria can't respire, they ferment. But fatty acids, if their fatty acids go into the mitochondria, they'll create tremendous oxygen radicals and kill the cell. So to protect the cell, they store the fatty acids as lipid drops. And what we did is we went through all major human cancers and we compared mitochondria structure and function with cytoplasmic lipid drops. Every major cancer has those two characteristics, and they're all fermenting, and they're all dependent on glucose and glutamine, regardless of what the mutations happen to be. All germline mutations, P53, Li-Fraumeni, BRCA1, all of these, every one of those genes damages number and structure and function of mitochondria, leading to a fermentation metabolism. So whether it's in the germline and the mutations, the somatic mutations are secondary downstream effects, and germline mutations cause disruption of mitochondrial function, leading to dysregulated cell growth as the result of the product of the inherited gene. None of them are 100% penetrant, meaning they can only be secondary risk factors. We have never found an inherited mutation in the genome that is associated with 100%. The highest is Li-Fraumeni, which is about 80%. But 20% of the people that have it never develop the cancer. So they're all considered secondary risk factors, just as it would be a viral infection, smoking, obesity, any of these. They're all secondary risk factors. The primary risk factor for the origin of cancer is a chronic disruption of oxidative phosphorylation coupled with a compensatory fermentation driven by amino acid and glucose fermentation processes.
Exactly. And I appreciate you explaining that so granularly, Tom. Jason, when we talk about insulin and obviously it being a growth factor, is there a relationship on hyperinsulinemia? I'm asking this, for a non-medical person, and mitochondrial health, and what are some strategies to either do or avoid when it comes to thinking about, and should we be thinking about preservation of mitochondrial health when it comes to metabolic dysregulation and hyperinsulinemia? And then even now I'm thinking, increased acidity with dialysis patients and their impaired ability to be able to, take care of that regulation that could potentially cause a metabolic injury to the mitochondria. Should we be seeing increased cancer levels in people that have impaired renal function?
It's not the acidity so much, but for sure, to get back to the enzyme, I think they work at slightly different levels because insulin is the sort of a hormonal level, whereas mitochondria is a cellular level. So they're not directly interactive. But I always think it's funny because it points to the importance of looking at the sort of bioenergetics of cancer, which is very striking because we spend so much time on somatic mutation theories, which is such a bad theory. Honestly, there's so much wrong with it that I'm surprised anybody still takes it seriously.
Jason, unfortunately, the National Cancer Institute takes it very, very seriously. That's why we have no progress.
No, that's what I thought too.
It's unbelievable. I agree with you 100%. I can't understand how rational minds can believe that. And yet, when you go to the National Cancer Institute website, it says cancer is 100 different genetic diseases. Unbelievable. Don't those guys ever read the literature?
Well, this is what I learned. So what I learned in the 90s in medical school was this two-hit hypothesis. That's what they're talking about back then, right? So you have,
Yeah.
it's not a genetic disease like polycystic kidney disease or sickle cell. You don't have one hit. You actually get one hit, then a second hit, and that's fine. That was the theory that I was taught, and probably a lot of people grew up on. But it's so completely wrong, because there are, you look at the average, cancer, and they did this. I think Bert Vogelstein did a study, and he took cancers and said, how many mutations are there, right? And it wasn't one, it wasn't two, it wasn't three, it wasn't four. The average breast cancer, I think, had like 50. And, some of the brain cancers that you see in young people, they're like 200 mutations, right? So it's like, okay, that's ridiculous, right? 50 different genetic changes, and you still think this is a viable theory? Like, how are you going to do that? Give them 50 different agents to attack all these different mutations? You can't do it. On the other hand, you look at bioenergetics, and every single cancer cell, I mean, not every, but almost all, share the same difficulty described ages ago by Warburg, right? That, hey, there's a real difference here in the way they generate energy, fermentation versus respiration. And why is that? And everybody says, well, with the somatic mutation theory, it's just a quirky problem that happens. It's just a mistake. It's like, what? So every single cancer in history makes the same mistake, and you think it's just a coinky-dink, right? It's like, what are you talking about? Clearly, this is at the heart of the cancer, is the bioenergetics. Whether you're talking about mitochondrial health, which is important, and it does influence it. So things like autophagy and mitophagy and stuff influence it. So when you do talk about intermittent fasting and stuff, it does influence it. But insulin and glucose, you're still talking about the same thing, bioenergetics, right? How do cells get energy? And I also think that it's always funny to me that they say, okay, so these cells produce lactic acid. These cancer cells produce lactic acid. And it's like, okay, and if that's just another big coinky-dink and it has no relevance. It's like, I don't think so. If every single cancer cell is producing lactic acid, it must be because lactic acid is good for the cancer cell, right? So basically, I think the cancer cell produces it. I think there's, they say, okay, it's not generating as much energy because oxidative phosphorylation generates way more ATP per glucose, right? Than fermentation. But that's only a problem if you don't have enough glucose. If you have a lot of glucose, it doesn't matter how many ATP you're generating per glucose because you have a ton of glucose sitting out there. So again, thinking about it that way, you say, well, okay, then why doesn't the cancer, if the cancer can choose either one, then it must be producing lactic acid for a reason. And I think it's a very good reason. The lactic acid protects the cancer cells. It breaks down, the cell membrane, so it allows it to expand. It protects it against attack from other, immune cells and other cells that want to destroy it, right? Because, your own body has a natural immunity, natural killer cells and stuff that try to attack it. So the lactic acid must be beneficial. And we need to look at why it's beneficial so you can attack it. But nobody looks at this. Nobody looks at these sort of really logical sort of questions and answers about how we can do this. They're all stuck in this. It's all about the genes. It's like, so you basically, you can't explain any of what's happened to these different populations, right? When you move a Japanese person from Japan to America, right? Their risk of certain cancers goes up tenfold, and, other cancers, gastric cancer, went down tenfold. Well, you can explain the gastric cancer, right? That was H. pylori probably, right? So it's like, okay, well, you're talking about environmental factors. What's the increase in risk of, say, breast cancer, right? And so the whole idea of genetics and somatic mutation theory, it's just like the somatic mutation. Like, there's so much wrong with it. Even in the New York Times, there was an article a few years ago talking about how they took esophageal cells from sort of, they took normal people, non-cancerous people, took these cells and just looked at them and compared it to cancer patients, like cancers. And they had all the same genetic mutations. It's the same. It wasn't any different. So if normal people are getting mutations but not getting cancer, then what's the difference, right? And that's where I think mitochondrial health is probably the underlying cellular sort of mechanism. And then on a hormonal level, which is a little bit higher because you're talking about hormones, not individual cells, then I think you're talking about, hey, what's the energetics? What's the glucose? What's the insulin? And also things like glutamine can be important, and triglycerides as well. So,
I mean, I'm sitting here thinking I'm like, there would be a study on, a high volume presentation, like just sprinkling a little sodium bicarb PO once a day PID. I'm sure you're thinking like, no, someday your kidneys are going to fix that. That's not going to do anything. But the point is, these are things that you want to think about and consider. And I think one good example that I believe you, Tom, had mentioned, maybe it was you, Jason, forgive me for not remembering, but there's just so much wealth of information about cardiac tissue. Like that is a nice way that I've been explaining since talking to you about how it's like, oh, this is a primary heart tumor, right? That in itself clues you in that the heart is the one tissue in your body that cannot have fatigue. So you could lift all day in the gym, you could do a 10 pound, a 50 pound, you could do more with the 10, but eventually the lactic acid builds up and you're just not able to continue. However, the heart is the only thing, and we don't talk about that enough, that doesn't, have that same phenomenon and is able to sustain for the entirety of your decades of life, God willing. And also, ironically, just doesn't seem to be that common of an organ to have a primary, cancer type on those myocytes.
Yeah, that's true. And neurons in the brain eat them. In order to have cancer, as Warburg said, you have to have a cell that can compensate oxphos inefficiency with fermentation. And cardiac myocytes and neurons can't do that for very long. They can do it over a short period of time if you were to have a heart attack or one of these kinds of things. But when you have a heart attack, you generally die from massive brain failure, even if you can get the person's heart back because the neurons are dead. They can sustain a massive upregulation of fermentation, a jolt for a short period of time, and you see massive amounts of lactic acid being produced. But it can't sustain that. One thing that's very interesting is when people do have heart attacks, the two things that you see massively accumulate in the blood are lactic acid and succinic acid. This is well known. And the succinic acid accumulates from glutamine fermentation in the mitochondria. It's called mitochondrial substrate level phosphorylation. The cancer cells are locked into this fermentation. They can't get out of it because their mitochondria are inefficient. When you accumulate the lactic acid in the you call the acidification. That's the result of mitochondrial inefficiency. So lactic acid, succinic acid accumulation in the microenvironment is the direct result of chronic mitochondrial insufficiency. That acidification is due to the fermentation of glucose and glutamine together. It also makes your immunotherapies, radiation therapy, and a lot of other therapies ineffective. They can't break through this acidification. So the question is, how do you reduce the acidification? You have to take away the glucose and the glutamine. We have not yet been able to find any cancer cell that can survive in the absence of glucose and glutamine. They can't grow on fatty acids. They can't grow on ketone bodies. We can't find anything that will, in other words, if you take glucose and glutamine and grow them on fatty acids, ketone bodies, anything you want, they die. And how do we know that? You know what we did? We took cancer cells from a range of human and mouse cancers, and we grew them in saline, just salt water, just salt solution, nothing. And we timed them with how long did it take for them to die? Mouse cells die quick because they have a high basal metabolic rate. Human cells took actually a few days for them to completely die just in saline solution. Can you believe it? Then we add stuff back one after another. What perks them up? What allows them to grow and survive again? And we did all 20 amino acids. We looked at a bunch of carbohydrates and stuff. And we found that it was glucose and glutamine were the only two major fuels that allowed those tumor cells to perk back up. If you throw a little glutamine, they get a little bit of burst. You put glucose and glutamine together, and it's unbelievable. And you have a few vitamins in there and a few other things. So clearly, we have interrogated the cancer cells to know precisely what they need to grow in a dysregulated way. And it's to two products driving the fermentation metabolism in these cells, making this not a complicated disease at all. And as Jason said, all the major cancers have the same problem. It's not like brain is different from bladder, different from breast. They all can't live without glucose and glutamine. So why are we focusing on thousands of different gene mutations when we're downstream effects and they have almost nothing to do with the dysregulated cell growth? Now, why doesn't somebody tell the NCI this? How is it possible that those guys sitting at the National Cancer Institute have no clue? They sit there doling out hundreds of millions of dollars in grant support for things that, and we got 1,700 people a day dying from cancer in the United States. That's 70 an hour, about 70 an hour. And everybody's chasing gene mutations that have almost nothing irrelevant to the nature of the problem.
I think,
Sledgy, tell me, what is wrong? What is wrong with this
I think it'd be like telling
picture?
on growing. I'm just kidding. I'm not going to put that
the
in the
original gene treatments were so good. The Gleevec and the Herceptin and stuff. They were really good, right? So then everybody thought this is the answer. I remember this because I was going through medical school at the time. It was like, oh, this is great. We're just going to find, this gene and treat it with this drug, which was like, okay, you have CMML, we can treat you with Gleevec because that's the gene mutation. It was so logical, right? And I think everybody got seduced by that they forget that after those couple of drugs, the first couple of drugs, there's been like 0 drugs in the last, 20 years now that have made that kind of a difference, right? Everything else, these all targeted gene mutations, they're not transformative, right? If you think about the way Gleevec transformed the treatment of CMML, it was like night and day. The thing turned on a dime, right? From a bad disease to like nothing at all, practically, right? It was a pill, you just took it. And that, so that's why it was so seductive, that the story was so seductive, the treatment was so seductive that everybody keeps looking. But if you haven't found it in 20 years, and you know how much money has gone into cancer research, well, it probably ain't there. Those were like the way outliers of the genetic paradigm. And they're just not true for most cancers. But the initial promise was so alluring that people are still going down this pathway, despite the fact that there's just way better, this whole evolutionary thought that it's an evolutionary disease. I mean, it changes everything, right? It changes how you think about the disease. It changes how you should approach the disease. Bioenergetics, like thinking about bioenergetics, because it's so core to all cancers, it just changes. Then you can start asking the questions, how can you affect it, right? Because we don't have great treatments, right? People say, can you starve cancers by just changing your diet? That's pretty broad, right? Because if you think about cancer cachexia, the cancer really takes what it wants from your body, right? Because when you lose weight, what you see in the normal non-cancerous is that you see that as you lose weight, your metabolic rate goes down because you have less and less energy stored, right? You don't see that in cancer cachexia. It's a totally different disease. The cancer just takes whatever it wants. It will completely catabolize your muscles. It'll burn out everything you have. That's why these people get so skinny. It's a different disease. It's not just weight loss. That's why people who say, oh, well, you should eat sugar and stuff so you gain weight. It's like, okay, this cancer is taking all your amino acids, breaking down your muscles, and you're gaining fat cells. Right? That's not a good strategy. And you're actually not even getting that much of it. So that whole idea of, oh, you should eat more, have some candy because you're losing weight, it doesn't actually pass any kind of logical thing. Like, that's third grade thinking, right? Like, we're supposed to be doctors, right? It's like you're operating at a level that's way too low, right? Because cancer cachexia is not just weight loss. It has nothing to do with normal weight loss. It's these cancers that are sucking all your energy out no matter what you do. So when you see, people losing weight and they're 80 pounds, their metabolic rate has not gone down at all. Their metabolic rate should have gone from 2,000 calories a day to like 1,200 a day, because that's what happens in every other person who tries to lose weight. That's the whole problem with losing weight, right? Is that when you reduce your calorie intake, your metabolic rate goes down because the body is trying to match it. That doesn't happen in cancer. Totally different disease. So again, pointing to the fact that bioenergetics, like how this cancer generates energy, which is, gets back to the power, the energy supply, which is mitochondria and stuff, is sort of core to the entire problem, right? And obviously we don't have all the answers, but at least it's a much more promising pathway than going on about genetics. And same as Tom, I'm always stunned when I go to any of these websites and it's a genetic disease. It's like the first sentence they say. It's like, goodness.
I know, it's bad. And I know you have hard stops. And Tom, I'm going to let you finish that out. I'm sorry. I want to ask on that point from both of you and your thought, Tom, please. Sorry. Speaking on that, if somebody had a malignancy, stage 2, stage 3, or is just like a strong family history and really worried, what are some more granular things to optimize? Obviously, like the insulin levels, mitochondrial injury downstream, in the way of antioxidants and berries, like even conceptually, what are the things, why does working out help? What are the things we can do to protect the much more rooted cause of what seems to be the, elicitor of all these mutations and neoplasms?
Well, let me just follow up on Jason's cachexia issue, which is really a core issue in cancer. And he's right. The cancer will take your muscles. The muscles begin to lose their composition. And what happens, the amino acids will go back into the bloodstream and go to the liver. And the liver will use it to make, through gluconeogenesis, you can actually make sugar from the dissolved amino acids from the muscles. And the glutamine itself is just a pure gold fuel for the tumor. And our work has shown that all metastatic cancers are of macrophage origin. A macrophage is part of our immune cell. They are the only cells that can actually move in and out. One of the few, they're part of the immune cells, move in and out of tissues. So you have two different kinds of cancer cells. You have the stem cells that grow like crazy, and then you have the metastatic cells. And they're all of macrophage. Every major human cancer that we have studied has macrophage characteristics. And they are the cells that are already programmed to enter and exit the bloodstream, this intravasation, extravasation. They're immunosuppression. And they'll lead to the cachexic phenotype and getting their energy from dissolving muscles. And Jason, it's very interesting when you're in cachexia, your insulin is high. When you go under calorie restriction, therapeutic fasting, your insulin and glucose go down. In cachexia, it's just a blown out opposite thing. You're right. So we have to look at this as a bioenergetic problem linked intrinsically to the mitochondria. And when you say, well, how do we manage? Well, if we know that the cells can't grow without fermentation fuels that are driving the dysregulated growth, then the strategy to manage cancer would be to simultaneously target the two fuels that are leading to this dysregulated growth. And they can't use fatty acids and ketones, as I've said. They store them. They don't use them. So when we look at calorie-restricted ketogenic diets or ketogenic metabolic therapy, we lower the blood sugar. And there's another very important thing. People always say, oh, you can never lower sugar enough to starve the tumor. That's not true. Because when you lower the blood sugar, what are the organs that take in the available sugar? It's the muscle. And there's you go back to your exercise again. Muscle does not share sugar. Muscle builds glycogen because if the beast is evolutionarily, if the bear is chasing you, you must run away. That comes from the energy. If your muscle shares glucose with other organs and the bears, you have no energy to run. So the muscle takes glucose out of the blood, stores it as glycogen for the muscle use. The brain is the biggest consumer of glucose in our body. So what little glucose is left when you lower blood sugar, the brain's going to get it. Who's going to get it last? The tumor cell gets the last. So you have to realize you're marginalizing their ability to survive. And then when you hit them with the glutamine targeting on the other end, which has to be done very strategically and carefully, you completely destroy the capability of this tumor to grow. You reduce the angiogenesis, you reduce the inflammation, and gradually your body, power to heal, will eventually go after and dissolve those tumor cells themselves and use that fuel for the energy for the rest of the body. It's unbelievable. It's called autolytic cannibalism, where the body will actually turn on the tumor and use the fuels for itself. Everything is related to bioenergetics. If you know why, and then we take poor patients and we treat them with all these terrible toxic poisons, reducing our own ability of the body to do what I just said. you compromise the body's ability to heal itself by the very treatments you're using to manage the disease. Makes absolutely no sense. Now, I'm not throwing out immunotherapies because they're based on the somatic mutation theory. They're based on an incorrect theory. But they could be a powerful tool if used correctly. If you can use a metabolic approach to shrink the tumor down to a few cells that are still alive, they must share something in common because they've survived together as a group. They may now be appropriately destroyed by a targeted immunotherapy, but not at the beginning, because at the beginning, you have all this hodgepodge, like Jay said, there's just a total hodgepodge of stuff. You may get some people to respond well, but you could also create hyperprogressive disease, not knowing the tool that you're using, making a bad situation much worse. So I think we just have to know how to use all the tools that we have in the appropriate way, with our understanding that this is fundamentally a metabolic disorder, a bioenergetic disorder, that we have to use the entire body's resources together with our understanding of what the foundational principle of what's going on inside the neoplastic cell itself. Once we understand this, the solution to the cancer problem will be obvious to everybody, and you're going to see drops in death rates like you can't imagine. But as long as the National Cancer Institute thinks it's a genetic disease, they ain't going to get any progress. It's just that simple. And the other thing that was, if you go back and look at the NCI history, you find in 1984, they invited the pharmaceutical industry to help the scientists of the National Cancer Institute find cures to cancer. That was putting the fox in the henhouse. How's that working out for us so far, right? We have more cancer today. Let me tell you something else, that smoking thing. In the early days of the anti-smoking campaign, get a load. They in 1991, they had this no more smoke, because secondhand smoke was bothering people. So we stopped smoking. Now, what they say is all the advances we've made since 1991, this giant 33% drop, was because we stopped smoking. The issue is that had we not smoked, we would have 33% more cancer deaths today than we actually have. So it's all smoke and mirrors if you want to know what's going on. It's unbelievable. Prevention was the only thing that really gave us some grip on the whole thing. It wasn't targeting gene mutations or any of that crazy stuff that seems to be the subject of millions of dollars thrown at these insane grants that are having no significant impact on survivability for cancer for the majority of people. It's, as I said, it's the greatest tragedy in the history of medicine, what we've had happen with this cancer fiasco.
Yeah. I mean, I agree. I have to leave in a minute, but I completely agree with you. I think that, using, understanding about glucose and amino acids and then trying to do sort of more fat-based therapies, to me, it's the only way, but to me, it's a very sort of, ham-handed sort of way to approach things. And I think that if we had more research, then we could develop much more sort of targeted ways. Like I know Tom, you had talked about hyperbaric oxygen and stuff, and I think there's still something in there, but it just hasn't been fleshed out, and nobody's thinking about these things. That it's such a promising pathway, that needs the research, because we're dealing with very primitive tools when we're just trying to sort of do this diet thing compared to, we're targeting this gene. We have so much money poured into the genes that we are so specific treatments of genes, and yet we know nothing, and we can't do anything about the bioenergetics, which sort of does it. So yeah, I mean, such a lost opportunity. So.
Well, that's predominantly because of the theory that's driving the industry. The somatic mutation theory is the theory driving research and development in the academic and pharmaceutical industries. And therefore, if cancer is not a genetic disease, it's a mitochondrial metabolic disorder, you're never going to achieve optimum outcome for the majority of patients. The theory is wrong, just as the geocentric theory of the solar system was not correct. Until you put the sun in the center of the solar system, all the planet movements made sense. As soon as you put the mitochondria in the center of the cancer problem, you're going to see major drops in death rates.
Jump.
For sure.
Sorry about that. It's great talking to you both.
I appreciate it, Gibson. When you mentioned the point about the acidity around the tumor or the lactic acid and how radiation isn't as effective, I'm really almost frustrated because I've never heard the next step. We hear about radio-resistant or radio-refractory tumors, and they become very challenging, especially when they're in a bad or stubborn place. Can we talk about that a little bit more on how, is there a way that we could potentially make a classically or traditionally radio refractory tumor more susceptible to radiation delivery, number one, and number two, how do we do that?
Well, I mean, it's very clear what's responsible for the acidification of the microenvironment in impeding radiation therapy and immunotherapy for that matter, and chemotherapy for that matter. And it's this acidification. What happens when you're fermenting? There's a P glycoprotein on the surface of cancer cells that as soon as the drugs come in, they pump them right out. And that's driven by a fermentation metabolism and causing the acidification. So what is that? As I said, we have interrogated these cancer cells, and it's the availability to convert glucose into lactic acid through the glycolysis cytoplasmic in the cytoplasm glycolysis pathway, dumping out lactic acid, and also glutamine dumping out succinic acid. So those are acids where the protons come out along with the lactate, the succinate, and you have acidified the microenvironment, thereby serving. It's not purposeful in the sense the cancer cells don't do this as a design mechanism to provide. They're doing it as a response to a failure in oxidative energy metabolism, but it also prevents other therapies from working. So if you were able to significantly reduce glucose and elevate ketone bodies, you then make these tumors extremely vulnerable to radiation therapy. You make them far more vulnerable to immunotherapies, and you can use half the dosage of chemotherapies and still have them have tremendous therapeutic benefit. So again, you have to remove that protection, that shield of acidification. Where is it coming from? It's coming from the fermentation of two fuels, glucose and glutamine. Simultaneous targeting of glucose and glutamine while under nutritional ketosis will allow radiation and all these other therapies to work infinitely better. Case, period. It's no question about that because everybody knows the acidification is blocking it. How do you get rid of the acidification? Where is it coming from? It's coming from the fermentation of two fuels only. Now, Sanjay, listen to this. No one anywhere is doing what I just said. They want to test drugs in clinical trials, double-blind crossover, one drug, this guy gets it, then we're going to do. That cannot be done. That kind of a trial must be booted out. Those trials have been monumental failures for a long period of time. Obviously, we had 1,700 people a day dying from cancer in the country. I mean, this is a tragedy. Why? Because we're testing drugs in the wrong way. It's a diet-drug combination that you're looking for to make the tumors vulnerable. The ultimate goal in all of this is how do we keep cancer patients alive longer with a higher quality of life? And it's metabolic therapy, man. That's what's going to do it. And you've got to target these fuels. And then you're going to find all these things that didn't work all of a sudden are going to work dramatically well under a different kind of. So we're not throwing anybody out of the boat. We're just telling them to reposition where you're sitting. You might not have been as important as you thought, but we still need what you're going to do. So so this is the thing. You're going to find dramatic results. I have no doubt about it. But what I'm talking seems to be too many issues to stand in the way, especially the theory. When the theory is incorrect, the outcome will never be optimal. And that's what we're experiencing right now.
Things like this podcast and what I hope to achieve even in the next 6 months, not for myself, but meaning like on transition of where I can put my bandwidth, that will hopefully really catalyze this. Because I know you have made it your mission. The man that is Thomas Seyfried you have spent now decades on trying to debunk this thing, and I cannot express my deep admiration for you for that.
Well, we need guys like you. You're going to be the guy. You're the guy in the trenches that's actually going to implement the paradigm change, because we have, when we go to oncologists and they never heard that glucose has any role in cancer and diet has no relevance, and that we're going to use all these expensive drugs. We have to re-educate the oncology community to know that the disorder that they thought was a genetic disease is actually a mitochondrial metabolic disease.
Well, Tom, I worry it's deeper than that. I worry that it's ultimately economic and capitalistic issue, which I know you said, but I really deeply think.
Well, then we have to give every cancer patient that comes into the clinic has to be given a little brochure to say, We thank them for their disease because it's a very powerful driver of the medical.
I know.
And nobody's going to say that. Nobody's going to tell cancer patients we're going to thank them because their disease is supporting a massively profitable industry. I think it should be patient outcome first and profitability second, not the reverse. But how do we do that? We have to have entrepreneurs who come into the picture and figure out how we can generate comparable profits from something that actually works and helps the patient. I don't think people are opposed to paying a large amount of money for something that actually works, but it's paying a lot of money that causes financial toxicity and death. This is a program that's not going to work. Eventually, people are going to wise up and say we can't do this anymore. It's actually a threat honestly, to national security, for crying out loud.
I honestly think, and I've never said this in my life before, I honestly think, in a weird way, if you have this beautiful big torch of passion and science and understanding, you have lit one for me that I now feel is like my primary mission. I've never felt in my core like, what is the primary mission? I just like do what the universe tells me. I feel like I want to help everything that you've done to really go full force on this very important issue. I'm going to say that in a second. I'm going to ask one thing real quick to put back right when you went on that about the acidity. And I'm afraid to ask this question, but I'm really afraid to ask this question. But is that why, and I could be wrong, liposarcomas are generally very chemo-refractory and even like radiation's not great? And is pancreatic cancer, which we know is classically a stubborn tumor, is that also an environmental? We know they hijack the immune system and hijack, extracellular matrix stuff. But do you think there's a role on the acidity of the microenvironment for these way more stubborn tumors that's reducing our hold on?
Yeah, well, certainly for pancreatic cancer and glioblastoma and some of the lung cancer, well, they're all like, driven with a macrophage component fused hybridization and these kinds of things, making them exceptionally dependent on both glucose and glutamine. The liposarcomas, on the other hand, they're not nearly as aggressive as some of these other more what we call malignant kinds of things. And I've always been interested in liposarcomas. They can come from certain kinds of mitochondrial abnormalities. I put that section in my book. When you have certain kinds of impacts on mitochondrial function, you get some kinds of these liposarcomas, and they can be a bitch to manage. They just hang on. It's just hack them away. Maybe surgery might be the only. But
it's like the cockroach of oncology.
No. They have an interesting, slightly different kind of metabolism from some of the more invasive malignant kinds. But that's kind of the interest of cell biology. And, I become less concerned about a liposarcoma than I become concerned by a pancreatic or a glioblastoma. These kinds of tumors, these are the ones that, and the ones that are lung cancer and breast cancer, the ones that are killing most of us is colon cancers and bladder and this kind of thing. But I mean, I did address the liposarcoma a little bit in my Cancer is a Mitochondrial Metabolic Disease paper, but I didn't do a deep dive on it. And
yeah, it's just unique because things don't work on it. And that usually happens when things are slow because tools we use are on replication.
The funny thing when you talk about liposarcomas, my sister many years ago had a dog with a liposarcoma, and we tried to use our metabolic therapy on it. The dog was given calorie restricted food. Usually it works on a lot of different kinds of malignant cancer. And what happened was that you had a liposarcoma on this dog. It was attached, and we put him on metabolic therapy. And then we found out it was a dog attached to a liposarcoma.
Oh my God!
It worked on the dog, but it didn't work on the liposarcoma. But that was an interesting N of one. But you're right. I think we need to figure that out a little bit more. I'm not claiming that we have a solution.
It's just so unique. Yeah,
compared to others. If we study a little bit more, we'll figure it out real quick.
It's funny you said that about dogs, because I had a gentleman from Harvard yesterday, Dr. Seung-Won Lim, and they found a workaround around their AI-enabled, tumor sensitivity workup stuff they do on live cells. And they started with dogs, one, because it kind of bypassed all of this, difficulty on, that's another whole nother podcast on even proving something that's utility because of all the red tape. But at least to blood cancers, dogs apparently have a much faster response to treatments, like to chemotherapy anyway. I don't know if there was a metabolic reason for that or if they just proliferate more or what, but apparently it's far more prompt and brisk than what we have to wait for with a couple of cycles in humans for things like Hodgkin's and other things.
Yeah. Well, dogs, I published that paper on the mast cell tumor on the dog's face, published in I think it was Frontiers in Nutrition or something. And that was the only thing we actually cured. We keep people alive very long, but who knows if they're cured or not. But the dog had complete resolution of the mast cell tumor using a metabolic therapy. And I have all the details in the paper itself. It's unbelievable. The dog died from old age from heart disease. So the cancer never came. That's the only way you know you're cured. If you live really old and you die from something other than the cancer, then you can say, oh, I was cured. Somebody say, but that's okay. If you're 98 years old, dropped dead of a heart attack when you had bladder cancer when you're 40, obviously metabolic therapy kept you alive for that long to die from something else. I mean, we're all terminal in one way or another. We just don't want to make it too premature. We don't want it to be premature. But you're right.
And I like that. That's what you say. You're like, look, I'm not claiming to cure cancers, but what I can tell you is we can have them in a deep, durable control. Just keep them, quote unquote, in check for a long time. That's the whole point. Our guy
from England, Pablo Kelly, he's had three debulking for his glioblastoma, IDH1. And he's just celebrating 10 years of survival now. He's married, 2 kids. He never had that. But his cancer is there. He's not cured. He just has it, and it grows slow, and he gets it debulked periodic. What do you have to have? You want to just live longer and have a somewhat of a normal life. I mean, be thankful, man. If you did standard of care, you would have been dead 9 years ago.
Right. And I mean, I'm laughing, and
I know
I'm the person that's doing this. Like I'm the one that's
it's like gallows humor, for Christ's sake. It's
yes, nailed it. Gosh, I really, if I can say I have a bromance love for someone, it is 100% you. Lastly, if you have the time, so sparing what I believe is also understated, gut microbiome health and its intimate relationship with the dexterity of the immune system, because I do believe having these narrow diets of just mac and cheese and chicken nuggets without the kind of heterogeneous stuff when it comes to different fruits and fibers, not the fiber itself, but fibrous things that have introduced kind of a dexterity to your immune system. Short of that, on a cellular metabolic level, when it comes to optimizing the health of our, mitochondria and cellular injury, I hope anyone can appreciate, one, in your discussions, the role and why we've been saying antioxidants for a long time, right? You do support, like that supports what you're saying. Oh, we know it helps radical ROS injury, etc. T's, like working out, what is that? It helps cardiovascular, processes. What are the things that make theoretical sense on someone that is trying to avoid a recurrence or prevent cancer to optimize this mitochondrial metabolic health that you were talking about?
Well, that's, the late Richard Veach from NIH. I had many discussions with him and George Cahill from the Joslin Diabetes Center. And when we look at what prevents reactive or oxidative stress, when you say antioxidant capabilities, well, water-only fasting and these kinds of things. When the body switches from glucose to ketone bodies, they're powerfully antioxidant. They actually improve the delta G prime of ATP hydrolysis within the mitochondria itself, which means for every breath of oxygen, for every mole of oxygen you take in, you actually get more efficient energy with minimal reactive oxygen species. So don't forget, we as a species evolved to always been in a semi-state of nutritional ketosis, where cancer was unheard of. Well, I'm not going to say we knew what was going on in the Paleolithic period, but we certainly know from modern humans that live according to traditional ways, they're generally in semi-ketosis. That's powerfully antioxidant. So, and you can supplement that. with your berries and things like this and certain things like that. So again, it's never one thing. It's a cocktail of things all go together to give you maximum metabolic efficiency, maximum homeostatic efficiency.
It's like a hypoxidant state,
Yes.
almost, if that's a word. So
Yes.
it's not. Yeah, you can kind of touch it every now and then, like pull out a weed here and there in your house with antioxidants. That's a bolus. But you're talking about just like a ketotic state.
Yeah.
A ketotic state can double over as a homonym
Yep.
to a hypoxidant state.
Yes, yes. And the other thing people are always talking about is their microbiome. They love this too. As soon as just mention microbiome, but let's keep our microbiome as healthy.
Yeah.
And then when you get cancer, you know what chemo does to your microbiome? God almighty. What are you talking about, your microbiome? And then you take some toxic chemical and you can't get off the pot.
And you kill it.
When your microbiome is screaming out, please, no more, no mas.
Right. No, it's
Makes no
true. And I
damn
think that's a factor.
It makes no damn sense. You're always talking about, then you give somebody this radiation and chemo and it knocks the crap out, literally knocks the crap out of your microbiome. I mean, when you think of the absurdity of stuff that we do to human beings in the name of trying to help them, it just makes no sense at all. But there's so many different ways you can look at this. And our goal has always been, how do we manage a chronic disease like cancer in a logical way based upon the hard science that we know is there? And all I have done is looked at the science. I'm trained biochemist and geneticist. We go back and you look and all of a sudden you can see all this thing and how wonderful it works and doesn't hurt people. And then you go out and you look and say, what are they doing out in the real world? What are you guys doing to these poor cancer patients? And
it's like we exist in two different worlds. It's like the biochemists and the Ph.D.s do something in some other country with a different passport, and somehow it's like get that and you trickle into another country and they see it off, and then they take what was done in this country when it's the clinical stuff now. And that's the problem, is there is just not a nice interstitching of that world. It's more of a handoff, and I think that's where things are lost. There's nobody in the middle that's actually kind of putting these things together like you are.
Yeah, well, that's the whole thing. Our work is directly translational, better than anyone else's that we can find. And the NIH, you got to do translational work. And then they build all these crazy models, genetically engineered cells and genetically engineered hosts, and you get all these things, and it just doesn't make a lot of sense. Everything has to be done with natural. As close to the real world as possible will give you the better outcomes. And translational, and almost none of this stuff is translational when you look at it. And they say, you cure these mice all the time, and you go to the clinic and you never get the. Well, they're not using the right tools. They're not using the right models. They're getting like I said in my book, when you bait the mousetrap with a picture of a piece of cheese, what you catch is a picture of a mouse. You don't really you don't really see what's really happening.
Yeah.
And what we're doing is we've just got pictures of cheese everywhere in our preclinical model systems.
I mean, the cheese is like the tumor microenvironment. We know that's important. That it seems to be a problem, and you're like, yes, and this is the cellular level and the kind of communication.
Yeah,
of this
we know why the microenvironment is screwed up. We just don't do anything about it, and we're making it worse in most cases. So again, you have to have a reevaluation of what's going on. You really need to. I don't know what the hell they're doing down at the National Cancer Institute. They keep backslapping and coming up with all this kind of crazy stuff, and people are dying out there. They're miserable, suffering that I can't even. I get all these emails, dozens, and sometimes I have to walk away. And I say, what are we doing to these poor people?
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
We're not treating them the way we should be treating them.
When you say "are" and "we," can you just tell us, anyone that's listening, that is driving and can't like Google you, what do you mean by us and we and our?
Well, I would say it's the establishment. What is the cancer establishment doing?
No, I mean for you, when you say what we're doing and what our focus is.
What our focus is to manage.
Who's our?
Well, the people that understand. Maybe you'll become one of these guys. I
think this podcast did it for me.
The guys who actually understand what cancer is and how it should be treated.
Yeah.
And then not being allowed to do that because of a standard of care written in granite. And standards of care should have never been written in granite. They have to be flexible. As new information comes along, we should be able to modify the standard of care. When we do clinical trials, you have the standard of care with metabolic therapy, and metabolic therapy by itself. Why is the missing control group never allowed to be done? This is the crazy stuff. So anyway, I have no law. I don't know how long it's going to take to break down the walls. But I'll tell you one thing: people want to live, and their motivation to want to live and know that there's an alternative to what we're doing may help change the system, and I'm hoping for that.
Dr. Seyfried, I really hope, I would love to shake your hand one day. I hope that's something you have time. But you live in Foxborough?
Yeah, I live in Foxborough. Right now I'm at the university at Boston College.
Gotcha. I really respect so much what you're doing. Have you met Greg Simon?
No, I haven't. I don't think.
The first cancer moonshot director?
Yeah.
If you have time one day, I would love to introduce y'all too. I honestly think y'all would be best friends. It's unbelievable how much you're alike. He was the first cancer moonshot director under Obama and Gore, and y'all are just so unbelievably similar. You appreciate the NCI shadiness, and y'all are just very, globally just kind of mission driven. He's a cancer patient. He's actually not a scientist. He used to be like a rock star, I believe it or not, but he's deeply respectable.
Yeah, well, I think once the theory changes and you realize that it's a mitochondrial metabolic disease, then everything will begin to change. Until that theory is, and all the textbooks say it's a genetic disease, so you're just perpetuating the lack of knowledge on the part of the industry. As you said, when you went to medical school, you never heard that cancer was anything other than a genetic disease. So clearly, the education in the medical schools have to change as well. So it's a big mission. But anyway, I'm here. I'm not going anywhere. We have a lot of stuff going, and all of our work is supported by philanthropy and private foundations. So not the national. They did it. When you do certain things, they're very, very valuable to support research. There's no question about it. National Institutes of Health has been a great resource for the nation. But right now in the cancer field and the cancer thing, it's just not working. And I think it can work once they realize the theory that they're working under is incorrect. And once they switch the theories, then you're going to see massive changes in the outcome and improvement for patient survival.
When we talk about like contiguous or cancers that, like in lung, smoker, or in colon, to Jason's point about the double hit, to me there it kind of makes sense, because it's like if you have a tubulovillous adenoma or an environment where there was one portion that's invasive and the other is not. That's the one challenge I can think about when it relates to the switch or a mutation, because what is it that makes something that's screwed up actually become invasive cancer versus pre?
Yeah, that's what we in my book on the origin of metastatic, in my paper, this is another thing that's very strange. I published a paper in 2013, I believe, on the origin of metastatic cancer, which talks about exactly what you just mentioned, why some cells are not invasive where other cells are invasive. And that comes from the fusion hybridization of our immune cells coming in. Cancer is viewed by the body as an unhealed wound, and it sends out signalings cytokines and factors that cause the immune system to come in and facilitate wound healing. And those cells are largely macrophages, and they throw out growth factors and cytokines, but it stimulates the stem cells to grow. Stem cells are not invasive. They grow like crazy, but they don't have the capabilities of metastasizing, intravasation, extravasation, this kind of thing. But when the macrophages fuse together with themselves and with one of these stem cells, you now have a cell that's not only dysregulated in cell growth, but also has the genetic apparatus to spread around and invade in a different way. Because when macrophages come out of your bloodstream as monocytes, they come right through the tissue to heal the wound, right? They're intravasation, extravasation. All this is macrophage behavior. This is exactly what metastatic cancer cells. But stem cell, one subpart of the tumor can't do that because they're mostly stem cells. So you can have this is why when you're looking at a cancer, you have a hodgepodge of different kinds of cells in that you have normal macs, you have fused macs, you have stem cells, you have a whole big, what they call cellular heterogeneity.
Fibroblasts.
Yeah, they're all in there. And the ones that blast out and become metastatic, every one of them has macrophage characteristics. I published all this. I showed everything. And that paper has been cited over 1,300 times. Now listen to this. They never read the paper. I only said in there metastasis is responsible for most cancer deaths, and that's the only reason they cite the paper without ever reading what I actually said. This is amazing. So the folks in the field aren't reading the literature; they're just taking little chunks of what this is. So I call it broken science. It's a problem where we have everything there to explain it, but people either are not reading it or don't understand it. So it's really, metastasis, why you have certain parts of the tumor, they grow fast, but they don't spread. The other part, little thing spreads all over the place. This is all due to the cell biology that exists inside that organ. A tumor is kind of an organ, its own blood system. Yeah.
So that cell is the cancer cell, or the even precancer cell, is the thing that eventually actually puts out the signaling to make that stem cell around it differentiate.
Yeah, well, it's not differentiated.
Environment.
Yeah, and don't forget, when you interrogate the metabolism of the stem cell and the metastatic cell, they're both fermenting. The problem is the stem cell does not have the genetic apparatus to allow it to penetrate local invasion, intravasation, extra. These are all very sophisticated biological processes. This just doesn't happen by chance. And the field calls it the epithelial mesenchymal transition, which is complete nonsense. But yet that's based on the somatic mutation theory. If you ever sit down and look at what the EMT, MET is, I have it in that paper, it's complete nonsense. And yet everybody is brainwashed into thinking about this. It's metastatic cancer is a macrophage disorder with macrophages fermenting with that, and they're fused hybrids has been shown many, many times. So you know what the metastatic cell is, driven heavily by glucose and glutamine. You know how to kill metastatic cells just because you understand their biology. Yeah, stem cells grow like crazy too. They'll kill you from mass effect by just being a big bringing in more immune cells. It's a situation of escalating biological chaos. All these different cells are doing what they're doing in the wrong context. So biology is now, the cells are screaming at each other in different ways, trying to figure out what's going on. And it's all things that were programmed into cells, but done in a completely wrong context.
Yeah, this is the first time ever of 2 years doing this podcast, every 2 weeks, that I've finally made a connection to what Michael Levin, Dr. Michael Levin, I don't know if you know him.
Yes, I know him well.
Yeah, he's doing what you're doing, basically, but not metabolic, but on the electric, bioelectric kind of, concept. This is the first time I've finally been able to make a connection, because when you said the tumor, I guess I knew, but I didn't know until you said it like this, is basically like a wound and it's trying to heal the wound and that entire process. One can now conceive Michael Levin's podcast on saying all that's happening is the cancer cell is actually just a regular cell that's doing what it's supposed to do, which all the cells have been doing since we were like protozoans, which is, once it believes it's autonomous, it will do anything to survive. That's what evolution has done inside of our bodies. So if you have this basically detachment from the electric or bioelectric harmony that in itself has its own communication outside of exosomes and signaling, with cells around the environment, then it's almost like, can you blame it for doing exactly what we're programmed to do when it now feels, foreign or autonomous?
Yeah, I don't like to give the cancer
a brain.
A brain. That's more teleological, which doesn't fit with our understanding of evolutionary biology. But when you say it's falling back, all the cells that existed on the planet before oxygen came into the atmosphere were fermenters. So the fermentation pathways are the most ancient pathways for energy. There was no oxygen. So when you have no oxygen, how do you get energy? And it's through substrate level phosphorylations, which are ancient pathways. And as I said, when you are fermenting glucose and glutamine, you're using substrate level phosphorylations. These are ancient pathways. And the cancer cells are simply falling back on these. And they exist in all of our cells in tiny little, they don't play a major role, but they're there. But in the cancer, they become dominant because the organelle that was controlling the differentiated state has lost control. Cells fall back on their default state, which is proliferation, driven by fermentation energy through substrate level phosphorylation. This is ancient pathways of energy metabolism that the cell, and they acidify the microenvironment, creating a world of difficulty for those trying to manage the disorder. But once you understand biological evolution and biochemistry, all this stuff to manage cancer should be done and can make a lot of sense and be very, very effective. Problem is most people don't know that or can't know. I don't know. It's just, I know it. I understand it. And I'm trying to tell the world that this is not an insurmountable problem. This is a very logical way to manage this disease based on your understanding of evolutionary biology and biochemistry. And Warburg had it right mostly, and we're correcting where he made his mistakes. Oh, he made a couple of mistakes, but we're fixing those mistakes. And we're going to come out with a new showing how Warburg's hypothesis merged now into the mitochondrial metabolic theory of cancer, and it will replace the somatic mutation theory of cancer. And then you're going to see death rates drop and people living longer and being healthier. And this is just the future. And you will be part of this future. Once you get to know this, your patients are going to love what you're doing to them and helping them out. But if the system allows you to do that, if the system doesn't allow you to do that, then you're going to be a very frustrated person.
Yeah. Tom, I appreciate it so much. I'm just always humbled, mind blown, mind rattling after speaking with you. This is, it's exhausting in the sense, in all the good reasons, but it's almost like it's a professional existential crisis. That's the way I would define it.
Well, I wish other members of your profession would have the same feeling.